On September 26, 2024, Michele Bergevin presented her PhD research on Cache Valley virus at the monthly meeting of the Community for Emerging and Zoonotic Diseases. Michele is a PhD candidate in the Department of Population Medicine at the University of Guelph. She earned a Bachelor of Science in Molecular Biology, Biomedical Engineering, and Economics at the Massachusetts Institute of Technology in the USA, and a Master of Applied Science in Biomedical Engineering at the University of Toronto.
All right, so
Great.
Michele, when you're ready, go ahead.
Okay. Hi, everyone. Thank you so much for having me. I'm really excited to be here today and talk about epidemiology of Cache Valley virus disease in Ontario, Canada, and what you want to know. But it being Truth and Reconciliation Week, I certainly want to acknowledge that the University of Guelph resides on the ancestral lands of the Arowandaran, the Anishinaabe, and the Haudenosaunee peoples in the treaty lands and territory of the Mississaugas of the Credit. I am very grateful to be able to share this beautiful land and water with people of the First Nations, Inuit, and Métis communities. I too strive to be a steward of the land and abide by the dish with one spoon creed, which guides how I live my life, how I raise my children, and how I approach my research. So first, just a brief overview to make sure that we're all on the same page. Cache Valley virus is from the genus Orthobunea virus. And Orthobunea viruses span the globe, with the individual species preferential to specific geographic regions. They replicate in and are transmitted by blood-feeding arthropods, and they're capable of infecting vertebrates. From a public and animal health importance, Cache Valley virus is endemic to North America and is part of the bunny and were serogroup. You may have heard of Jamestown Canyon virus, lacrosse virus, and snowshoe hare virus, which are also endemic in North America and part of the California serogroup. So in the Orthobunea virus genus include Schmallenberg virus, causing a horrific disease in livestock in Europe, Akabane virus, similar outcome in Australia, and what we've heard a lot about recently in the news, Oropouche virus, part of the Simbo group in Simbo serogroup and endemic in South America. So the rationale for this work resides in the fact that the true public health burden of Cache Valley virus disease is unknown in Canada. And despite these viruses likely being unfamiliar to the average person, the Public Health Agency of Canada ranks them as a top priority among vector-borne diseases in a climate change context. And that's really for three main reasons. It's zoonotic, causing disease in humans and animals, particularly livestock, specifically sheep predominantly. There's no treatment and no vaccine in either animals or sheep, or animals or humans. And geographic expansion of CVV is anticipated due to a warming climate. And since the audience is likely familiar with West Nile virus, I wanted to mention a couple studies that reassessed suspect West Nile virus cases in Canada a number of years ago. And I reported that actually 28% had been infected with Jamestown Canyon or snowshoe hare virus, and 7% were infected with Cache Valley virus. And these results motivate the question, does Cache Valley virus warrant more attention in public health? And by actively assessing the epidemiologic data, are we just scratching the surface of a potentially much bigger health problem for Canadians? And that motivated a lot of what my PhD research has been about. Okay, so in terms of the transmission cycle, the sylvatic cycle involves infected mosquitoes taking a blood meal from a reservoir host, which is one that can amplify or maintain the virus. In the case of Cache Valley, that's the white-tailed deer. And that would be the horizontal transmission pathway. It can also be passed vertically from an infected female to her offspring, and during venereal transmission when adults copulate. Epizootic cycles occur when infected mosquitoes take a blood meal from an incidental host, including humans and sheep. And in this case, insufficient viral loads occur such that subsequent or naive mosquitoes taking subsequent blood meals cannot become infected. But disease can manifest in these incidental hosts, and that's why they're often called dead-end hosts. So Cache Valley virus antibodies have been detected in 22 mammalian species, but their roles in transmission are not clear, namely because it's just underreported or understudied. In terms of mosquito species, the virus has been detected in over 40 species across 7 genera, and the primary vectors are not known. But what's been guiding scientists in which mosquito species may play a predominant role is determined by the amount of virus detected through viral loads, the frequency of detection over space and time, and vector competency studies, as well as blood meal preferences. And I will talk in a little bit about 7 species in particular that we believe are playing a role in transmission because they check off, a lot of these boxes. And also another important thing to be aware of is that longitudinal studies from the US, specifically in Connecticut and New York, have identified that while mosquito abundance is declining late in the season, that's when actually the Cache Valley virus loads are at their peak, particularly in September and October. All right, my slide. In terms of geographic distribution, as I mentioned, Cache Valley virus is widespread across the US and Canada, having been detected in 5 Canadian provinces and 33 American states based on peer-reviewed literature and gray literature that conducted mosquito surveillance, seroprevalence in vertebrate species, and disease case reports in humans or livestock. Disease is, well, when humans become infected, typically they're asymptomatic or have mild and self-limiting symptoms. On rare occasions, 7 to be precise, horrible disease has manifested. And this really nice table came from a recent study just summarizing the severity of disease, in which case 3 out of the 7 died. 3 had long-term sequelae that greatly were debilitating, and then 1 actually just wasn't fully recovered after being released from the hospital. In terms of sheep in particular, these are images of classic signs of disease. So the image on the left or middle illustrates the hump spine, the twisted limbs, and the fused joints. And the image on the right shows the classic curved spine. Mummification can occur, and an underdeveloped brain can occur. So the virus is targeting central nervous system and musculoskeletal system. And oftentimes when sheep producers or veterinarians see these classic signs, they don't actually submit samples. They just conclude that it's Cache Valley virus disease. And that is one of the reasons why we believe that, the disease is underreported. And abortion or stillbirth can occur depending on at which phase in gestation the fetuses are infected. And so just to illustrate, if a sheep is infected, it typically will be asymptomatic and it will clear the virus and develop long-term antibodies. However, if it is infected when pregnant in the first trimester, the virus can cross the placenta and infect 1 or more of the fetuses. And if this happens within, days 0 to about 28, abortion typically occurs, manifesting as infertility issues in the flock. And if it occurs in around days 28 to 50, then severe congenital defects occur, such as the images that you saw. And the dam will develop these long-term antibodies, and fetuses infected beyond the first trimester will also be able to clear the virus and have antibodies when they're born. But the antibodies cannot be passed from a dam across the placenta to the fetuses. And so on the rare occasion when newborn lambs are born with the disease, they will die within the first few days of life. And in terms of detecting it, you are unlikely to isolate the virus because it has been cleared during gestation. So the common way to conclude it was due to Cache Valley virus is to test for antibodies prior to any sort of colostrum intake. And then real quickly, certainly common in Ontario is this breeding schedule where sheep are bred during the latter part of the mosquito season to ensure certain weight points are hit in lambs in the new year. to achieve market demand, but that puts them at great risk of developing disease in the flock in CVV endemic regions. And this is why we tend to see disease newborn lambs in the December-January timeframe. And this is just illustrating from one of our scoping reviews that the virus is detected. This is just representing citations, not the amount in the population, but illustrating that the disease is commonly detected in the late part of the mosquito season, predominantly August and September. And so one of the two research chapters I wanted to talk to you about today relates to mosquito vectors, specifically in Ontario. So the image on the left, which I've talked about, all the factors that go into maintaining Cache Valley virus transmission in its sylvatic cycle, we kind of took it apart and looked at it through different lenses. And so this first chapter looks at the vectors themselves. And so we wanted to identify spatial and temporal patterns in mosquito species implicated with CVV transmission based on longitudinal surveillance data in Ontario. And so we obtained surveillance data from 2002, when the program started here in Ontario, to 2020. And the surveillance was put in place, namely to monitor West Nile virus and Culex species. But nonetheless, it provided a starting point for us to look at the vectors. And so here on the left is just showing the geographic boundaries of the health units. And the surveillance was actually conducted at the health unit level. And so there might be some, while the protocol was the same across the board, execution of surveillance may vary across health units based on available resources and regional health priorities. And then the image on the right is just a summary of all the mosquito traps over the 19 years. And you can see concentration in the southern regions and also in urban environments. And we specifically looked at these 7 mosquito species: Aedes vexans, Cochleartidia perturbans, Oculoritatis trivitatis, Oculoritatis canadensis, Anopheles punctopenis, and Quadrimaculatus, and Aedes cinereus, because those were the ones that have most frequently been reported as being positive for Cache Valley virus in surveillance in the US. There has been no surveillance of mosquito species, certainly in Ontario. And so this was one step towards emphasizing the importance for it, the need for it. And we see here that of the 4.5 million species that were identified over the 19-year period, 65% represented Cache Valley virus vectors. And they were predominantly Aedes vexans and Coch perturbans, each of those showing different trends over the 19-year period, with an overall increase in abundance with Coch perturbans and a decrease in Aedes vexans. The remaining 5 were mostly stable from year to year, and the color, you can see the different trends. So next, we were interested in identifying emergence and decline patterns in each of these 7 species. So this is from a provincial standpoint. And the takeaway with this figure is just to see that, number one, these species, certainly Aedes vexans and Anopheles punctopenis, which are the first to emerge, have arrived at the beginning of surveillance season. And in fact, we're likely not even capturing true emergence because they're being detected in the first week of surveillance. And same thing is going on the backside, where we are detecting these species in the last week of surveillance. So surveillance in this image isn't sufficient to truly validate emergence and decline in these species. Looking at this data through a different lens, through seasonal distribution of the vectors, they are ordered from top down, from earliest emerger to latest. Again, you can see the dominance of the Aedes vexans in the second to bottom column of the Cochleartidia perturbans. They have different growth patterns, but in the right column, I have tailored the y-axis so you can really see the abundance peaks and different growth trends across the 7 species. The pinkish red panels in each, or bars in each of the panels, is just highlighting the latter part of the season when Cache Valley virus is most detected. So we do not have virus data. We only have the vectors in this study. But what we do see is that if you were to aggregate all these 7 species represented in the bottom row, that there's still a lot of CVV vector activity late in the season, which is supported, it illustrates that Cache Valley virus can be supported through the season from May till the end of October. And then this image is just illustrating the variance in surveillance across health units, because your takeaways are only as good as the data. So first, we wanted to truly understand the quality of the data from the lens of evaluating Cache Valley virus vectors. And so you can see quite a bit of variability in the duration of surveillance seasons. This is just representing the number of weeks sampled each year. And the red line indicates the average of 14 weeks sampled across the 36 public health units. And I just circled the Northern Health Unit, PQP, because you'll see in the next figure that while the surveillance season was shorter, relatively speaking, than a lot of the Southern Health Units, you can see that it was not, or it did not correlate with the number of traps used per week. So in the PQP health unit, they did have above average number of traps within the weeks, even though fewer weeks were sampled each year. Again, the top row is illustrating that the most traps, as we saw in that earlier image, are in the Southern units. And so this is just a quick snapshot to show the number of traps out there each week of each year. And then looking at the data, again, through an aggregate level, we broke it out by public health unit. And you can see here clearly that even within most of the health units, there is aggregated CVV vector activity through the end of the season, again, supporting sustainability of virus in endemic regions. And then the takeaway with this image was the final point about how much we can conclude from the surveillance data. We can certainly capture peak abundance across the health units among the 7 vectors, but we are still missing the true emergence and decline. So if you were to just look at this data from a seasonal perspective, in the lower right image, you see trends that you would expect from from the vectors. But in the top left image, you can see that as soon as surveillance started, again, we're picking up signal of activity among these vectors. And so how early is it starting is important to know, and how late is it persisting into the season to be able to truly assess the risk period. And then the final figure for this chapter is just looking at the average seasonal abundance of CVV vectors across health units from a spatial perspective. We can see even when accounting for more traps by averaging all traps within a week, we see a concentration of activity in the Southern health units, and the lower right is an aggregate perspective. And so the significance with this is twofold, really. Seasonal activity at the aggregate level of CVV vector species has been shown to persist for the surveillance period of about 5 months, from mid-May to mid-October, and likely supports the enzootic cycle of CVV in Ontario. And relatively high seasonal abundance of CVV vectors in the Southern Ontario coincides with the large populations of susceptible human and animal, including livestock hosts, which necessitates targeting CVV surveillance for mosquito and animal populations. And limitations, I've already mentioned that we're working with the surveillance data that was optimized for detecting West Nile virus and Culex species, which tend to occur in more urban environments versus Cache Valley vectors, which tend to be in more agricultural and rural settings alongside woodlots. And then also that the surveillance illustrated in certain health units at certain years, we weren't necessarily capturing true emergence and decline, but indeed we were able to illustrate peak activity and activity throughout the season. Okay, so real briefly, I'm just going to highlight this chapter focused on the work with respect to sheep. It is published, so there's a way to get this additional data. But the takeaway here is that in recent years, we've been hearing about sheep outbreaks, particularly in Ontario. And the top left was the first outbreak that drew our attention to the fact that Cache Valley virus is pathogenic in vertebrate species. And so you can see the impact in this flock from a typical less than 3% infertility rate, less than 5% lamb mortality, to 10% infertility due to the outbreak, and greater than 6% lamb mortality and severe congenital defects, up to 19%. So it's these big impacts that usually draw attention and motivate veterinarians, sheep producers, healthcare practitioners to start testing to see the cause of these clinical signs. And so there's no surveillance work in Ontario among sheep. And so we set out to conduct a cross-sectional study that determined the prevalence of CVV seropositivity in sheep across Ontario, as well as assess farm management risk factors associated with CVV seropositivity using statistical modeling, since that was something that might be manageable and manipulated to reduce risk. And so, excuse me, we targeted the 5 sheep districts with the largest ewes, and we recruited 18 farms with 20 mature ewes at each farm. And a mature ewe was defined as having given birth at least once. And then we performed a virus neutralization test to assess seropositivity. We also conducted a questionnaire to gather information on what we perceived as potential risk factors, which addressed flock demographics, breeding schedule, shearing schedule, housing, medical history, environmental landscape, and mosquito prevention practices. And then we performed a mixed logistic regression with a random effect for farm, looking at these different factors. And I just wanted to give a snapshot of kind of the demographics of the sheep. So fairly evenly distributed in terms of shearing schedule and flock size. On the far right, we did verify that the majority of sheep sampled had been on the farm for the duration of their life to ensure that what we were testing was representative of the exposure in Ontario. And ultimately, we had 364 samples, and the average age was 3 years, 7 months, but spanned from 1 year and 2 months to 10 years and 4 months. So in terms of seroprevalence, we see that it is widely distributed across the farms. And the 2 where we did not detect activity were interestingly near each other. Also, in terms of age, it's not surprising that the older the ewes were, the more likely they were seropositive, simply because they've been exposed for a longer period of time. And of the 2 farms where we did not detect virus, they came from large flocks. But unfortunately, the ewes tested on both those farms range between 1 and 2 years of age. And so it's interesting to think if we were to have sampled older ewes, would we have come away with different results? We won't know. But obviously the clear trend with age. And then one other thing I wanted to mention was the incredible variation in housing, which is unfortunate given we think that there's a big association with exposure to infected mosquitoes. And so the point here is you see a lot of color on both pie charts, indicating that there was a lot of movement both across farms and among ewes within farm in terms of the different housing. It depended on the time of day as well as the life stage in terms of breeding. And so this big table, just the takeaway is that the significant risk factors that were found to be associated with CVV seropositivity included increased age, being housed near wetlands, lakes, or ponds, so natural bodies of stagnant water, and then also flock size. But we do feel that flock size was likely a proxy for another risk factor, possibly housing. And certainly future work would need to include a larger sample size in terms of number of farms to be able to perform additional analytical research on interaction terms. Also, you can see that most of the variance was actually at the U level, which again is not surprising given the incredible variation in terms of housing based on stage and breeding. So I think I've pretty much covered this, but in conclusion, CVV is prevalent across Southern Ontario. 1 in 3 ewes were seropositive, and 9 out of 10 flocks were seropositive. And ewes housed near sources of natural stagnant water, increased age, or part of a small flock have increased likelihood of being seropositive for CVV in endemic regions. But flock size is likely a proxy for another risk factor, possibly housing. And intervention tactics will have greatest impact at the sheep level, unsurprisingly, given the wide range in farm management practices that varied with life stage. So the significance from this work is that outdoor housing during mosquito season and age are positively associated with CVV seroconversion, which suggests that in early years, ewes bred outside the mosquito season and housed outdoors during the mosquito season, if not breeding, may reduce the risk of CVV disease in the flock. And some limitations I touched upon. There was great variability in housing across farms, and to address that, we would really need to do further work with a larger sample size of farms. And there was minimal age variance among the ewes at the 2 farms where we did not detect virus. And landscape risk factors were subjectively determined, and future work would certainly benefit from a more objective means to account for land use. And so this final slide is just looking at, my wish list of next steps, being able to, number 1, perform a cohort study so that I could really hone in on the different housing. Also, increased mosquito and vertebrate surveillance around sheep farms, as well as virus testing, and incorporation of objective weather and land use metrics to assess impacts. You can see in this figure, this is showing the positive flocks in red, the blue flocks where we did not detect virus, and the yellow circles indicate traps in the year that probably indicated exposure to those sheep farms. And there's not a lot of yellow dots around where the farms were. So again, why I think there'd be a lot of value in doing further surveillance work on site at the farms. So a big thank you to all my collaborators. And if you have, I certainly welcome continued feedback and questions. Please write down my email address and reach out to me if you have any questions or want to talk further. So thank you.
Great. Thank you very much, Michelle. That was very interesting. So we do have some time for questions. So there's one currently in the chat. So CFIA is doing some work on climate change, and Eric is wondering whether Cache Valley virus should be identified as a disease whose prevalence and distribution is impacted by climate change.
Absolutely. I'm not biased or anything, but there was an interesting study that was published this year Muller, M-U-L-L-E-R, just look up Cache Valley virus 2024, and they did looked at it in the context of climate change and illustrated that they do anticipate expansion of both the vectors and the vertebrate hosts. But what was really interesting about that study is they essentially also used AI to review the literature and extract key species that might play a role, because again, there's very little known about other vertebrate and vector species driving transmission. And I think as a result of the bias in the data that we all use for certainly vector analysis and the limited data with maintenance host surveillance, Culex was identified as one of the mosquito species to drive transmission. And that was really just on frequency of identification in the literature. And so the point is that it's just really important that we also complement any sort of like climate research with additional surveillance work that is optimized for studying those perpetuating CVV transmission.
Great. Yeah, thank you. We have another one here in the chat. So did the farms that were seropositive report current or previous congenital abnormalities in their lambs?
Great question. Yeah, I do have that data, and some of them did. A large number had said, well, a portion had said that they've never experienced disease, but they did have seropositive flock, but they also did not breed their ewes during mosquito season to suggest that they were avoiding the risk period for disease. But I would say about 50% had indicated that they had signs of disease in their flock. And of the 2 that we did not detect virus in, they also said that they did not experience disease in their flock. And I should also mention that there was a very interesting sero survey done in response to the outbreak in 2011-2012, performed by Dr. Paula Menzies and Shannon Meadows. And it was never published, but they were collecting data for blue tongue virus or Q fever assessments. And they had samples on hand that somewhat overlapped my regions. And this data was from 2010. And 21 of the 26 flocks were seropositive. And the one region where they definitely saw seronegative flocks was again in the similar region where I did as well. So from transitioning from their work to mine, which was over 10 years later, it seems like it's spreading and there are pockets that haven't been touched yet, but this is a bit speculative, so.
Okay, interesting. Yeah, thank you. Are there any other questions from anybody on the call? Okay, well, if not, oh, I see one from Jen. Go ahead.
Sure. Hi, everyone. Sorry, I'm having computer troubles today. It seems to be very unstable. Hi, Michele. Thank you very much for your presentation. That was super interesting. I was wondering, in your slide where you mentioned that 65% of mosquitoes were considered vectors, is that 65% of species or individual mosquitoes? And then is it that they've been demonstrated as competent vectors, or they're just kind of co-located with the disease and had viral identification or viral load?
Good question. So that was the 65% was just of the total that had been identified. Then we were focused on those 7 species. So it's not ruling out others, but of those 7 species, they represented 65% of the total 4.5 million mosquitoes over the 19 years. And we identified those 7 species from work was twofold. One was this classic study from 2014 out of Connecticut, where they had longitudinal data and they tested virus. and they indicated high frequency of detection, both over space and time. It was 4 detections in 7 years, so still somewhat minimal, of those species, as well as competency having been proven in the majority, but not all, because a lot of vector competency studies have been focusing on more exotic species like Aedes albopictus and Aedes aegypti and the Culex species for West Nile virus. But a number of them have been demonstrated as being experimentally competent, and all those species are preferentially feed on mammals, mostly white-tailed deer, and have been reported in humans. Also, just the frequency in the literature of having been detected as well, even if it was just one, just being recognized in that piece of literature also guided our decision to focus in on these 7 species. So we also probably would have been interested in Aedes albopictus, but there's not much data in terms of surveillance in Ontario yet. So
thank you. That's really interesting. And for the person who's getting bit in their backyard, is there like an easy way to identify what your mosquito looks like?
Yeah, I don't know. I'm pretty sure I got Cache Valley virus yesterday walking home at night through the Arboretum, because it's definitely that time period. I don't know. I would like to know, but that still remains to be seen. So all we can hope is that if it is being passed, we're mounting an immune response and we're being protected. It's really those that have the weakened immune systems that have to be careful.
Thank you very much.
Great, thank you. So there's one other question in the chat here from Cynthia. Are there any estimates on the financial impact?
I think about that a lot. The short answer is I don't have a good answer right now. I do know that certainly in flocks here, it's impactful financially and emotionally. I mean, the shepherds seem pretty traumatized from having gone through something like that, and it affects how they manage their sheep moving forward. Also, the dams can experience dystocia, which can affect their performance moving forward. And one thing that came up in my research was that if shepherds are seeing defects in newborns, it could sometimes be misinterpreted as something genetic, and that really affects purebred flocks, because then they're at it's jeopardizing their risk and basically their product. So I don't have numbers tied to that, but I do know that greatly impacts. And wouldn't it be nice if there was a snap test to go with like Metavissa, where you could test for exposure to Cache Valley virus, and once you know that they have antibodies, they're no longer on your radar. Otherwise, just having to manipulate breeding schedule, because a lot of sheep producers now are going with this accelerated program, and that's increasing the risk if they happen to be breeding these naive ewes. So yeah, but I don't have a good answer, but it keeps me up at night, so I should work on a number.
Great. Well, thank you, Michele. Yeah, this has been really good. I think bringing to our attention kind of a disease that maybe goes under the radar a little bit. So thank you.